Baltimore, Maryland, August 3rd, 2023. A woman picks up her phone and opens an app she's had for 2 years. She sees a notification she doesn't understand at first. A possible relative has been identified. Catoctin Furnace Cemetery, Maryland, late 18th century. She has never heard of Catoctin Furnace. The person she has been matched to has been dead for more than 200 years. She sits down. What made that notification possible was a study published that same morning in one of the most prestigious scientific journals on Earth. Dr. Eodoin Harney, a geneticist at 23andMe who trained in the laboratory of Dr. David Reich at Harvard, the most cited ancient DNA researcher alive, had spent years on a question that most scientists had told her was unanswerable. She wanted to know if it was possible to extract whole genome data from the bones of enslaved African Americans and match that data to living people. Not approximate them, not estimate regional connections, actually match them, individual to individual, across 2 centuries of silence. In August 2023, she published the answer. Yes, her team recovered genomic data from 27 people buried in an unmarked cemetery at a Maryland iron forge called Catoctin Furnace, men, women, and children who lived and worked and died there between 1774 and 1850. They cross-referenced those ancient genomes against 9.2 million living people in the 23andMe database who had consented to research participation. And the results came back. 41,799 living people shared DNA with someone in that cemetery. Think about that number for a second. Not dozens, not hundreds. 41,799. Each one descended measurably, specifically genetically, from a person who had no name in any official record, no census entry, no birth document, no grave marker with text. The people at Catoctin died before 1870, and 1870 is the year the United States Census first listed Black Americans by name. Before that date, enslaved people were property. Property does not get a name in a ledger, it gets a number. Genealogists call what lies before 1870 the information cliff. For most African American families, the story simply stops there, falls off the edge. Until now, no scientific instrument existed that could reach below that cliff. The Catoctin study was the first proof that one could. Stay with me, because this next part is the reason this video exists. What this means is not just genealogical. It means that the DNA held inside the bones of enslaved people, buried for 2 centuries in unmarked graves across this country, contains recoverable, specific, individual-level information about who these people were, where they came from, and who is alive today because of them. That is not a metaphor. That is a data output. And if it worked at Catoctin, it can work anywhere in the United States where enslaved people were buried, which is to say, nearly everywhere. If you've ever actually wanted to understand where a people come from, not the simplified version, not the family story, but the real forensic answer built from bone and genome, subscribe now. Because what comes next goes further into this than most people are prepared to go. Among those 27 individuals at Catoctin, researchers found something unexpected. Their ancestry did not match what historians believed it should be. A lineage that should have been absent from that cemetery, according to every prior assumption about who was enslaved in the Chesapeake region, had survived in that soil for 200 years, waiting. Before we go any further, drop in the comments if you've ever taken a DNA ancestry test. What was the most unexpected result you got? And if you have, why not? Because both answers are going to matter for where this is heading. The ancestry that came back from Catoctin did not point primarily to the Igbo or the Yoruba of Nigeria, the groups most associated with African American ancestry today, the names most commonly returned by consumer DNA tests to living Black Americans. It pointed somewhere else. The Wolof and the Mandinka of Senegambia, the Congo of Central Africa, what is now Angola and the Democratic Republic of Congo. These were the genetic signatures dominant in the Catoctin cemetery. And to understand why, you have to go to a historical database that most people have never heard of. Professor Henry Louis Gates, Jr., co-author on the Harney study and director of the Hutchins Center for African and African American Research at Harvard, pointed his colleagues to the Transatlantic Slave Trade Database. It is the largest historical record of slave ship movements ever compiled. And according to that database, approximately 25% of all Africans transported to North America came from Senegambia. Another 25% came from Congo, Angola. The Chesapeake region where Catoctin operated drew heavily from both corridors in the late 18th century. The DNA matched the ships. 6 words. But think about what they mean. Every haplogroup in that cemetery was a shipping route. Every maternal lineage was a port of departure. The genetic analysis was not just recovering personal history. It was mapping a supply chain. Reading the logistics of one of the largest forced migrations in human history through the bodies of the people who were moved. But here's where it gets strange, because that match, DNA to shipping records, is actually the easy part of this story. The Wolof people of Senegambia were, for centuries before the trade, a sophisticated empire: traders, scholars, warriors, farmers. The Congo people had built one of the largest and most politically organized kingdoms in sub-Saharan Africa, with a legal system, a diplomatic corps, and trade networks stretching across the continent. These were not people without history. They were people whose history was stripped from them with such thoroughness that their American descendants, 2.5 centuries later, required a machine to recover the names of the nations they came from. But this is also where the data reveals a problem, and the problem is bigger than the Catoctin cemetery. Among the 27 individuals at Catoctin, there are people whose African origins cannot be traced to any specific surviving group, not because their DNA is poorly preserved, but because the populations they descended from no longer exist in a form that any genetic database can recognize. An entire migration wave, an entire people present in the genome and nowhere else. Those missing origins, those people who cannot be traced to any surviving African population, are not the exception. They are a warning about what the slave trade actually did to Africa itself. Dr. Theodore Schur of the University of Pennsylvania and his colleague, Dr. Raquel Fleskes of the University of Connecticut, documented this in a parallel ancient DNA study published in the Proceedings of the National Academy of Sciences in January 2023. They analyzed 36 African-descended individuals from an 18th century burial ground discovered during construction in Charleston, South Carolina, the largest collection of ancient African-American genomes analyzed up to that point. Some of the Charleston individuals didn't match any contemporary African reference population with statistical confidence, not because of poor DNA, because the populations they descended from had been fractured by the trade itself beyond reconstruction. The reference database we used to identify African ancestry was built from the survivors of the same catastrophe we're trying to measure. Hold on to that, because it's about to mean something completely different. Because inside that damaged archive, inside that cemetery of unnamed people in Maryland, the researchers found something that nobody expected. They found families, not just individuals, families. Of the 27 people at Catoctin, 15 could be grouped into 5 distinct biological family units, confirmed by genomic relatedness analysis: mothers and their children, siblings. And when the researchers mapped where the related individuals were buried, the bones were near the bones of the people they belonged to. In a cemetery where no authority had any legal reason to arrange families together, the families were together. Think about what that means. American slavery legally prohibited enslaved people from entering marriage contracts. Their family relationships had no standing in law. They could be sold, separated, and relocated at any moment. And yet in that hillside in Maryland, the families had held. The assumption that the intimate structures of enslaved family life are permanently unrecoverable, that the legal erasure of black family bonds also erased the evidence of their existence, that assumption is wrong. The genome kept the record. 5 families, mothers and children, confirmed. But here is what nobody was ready for. Among all 15 related individuals in those 5 family groups, not a single father-child relationship was found, not one. Every family is a mother and her children. The fathers are completely absent, and the reason is not grief. The reason is something the data makes impossible to look away from. The fathers at Catoctin are absent from the African family structures because their children's chromosomes do not point to Africa at all on the paternal side. They point to Europe. But before we go there, because we have to go there is something else in those genomes that tells a completely different kind of story. The same team at the Smithsonian noted the presence of the sickle cell variant in the Catoctin genomes. One specific, ancient mutation in the hemoglobin beta gene, designated HBB beta S, and what that mutation represents is not a medical liability. It is 20,000 years of survival intelligence encoded in blood. The sickle cell mutation evolved in West and Central Africa as a defense against Plasmodium falciparum, the most lethal form of malaria. Carry one copy, and you are significantly protected from the parasite's worst effects. The populations with the highest frequencies of this variant, the Wolof, the Congo, the Yoruba, the Igbo, are precisely the populations most heavily targeted by slave trade networks. Because they lived in the most agriculturally productive, most densely settled, most malaria-endemic landscapes in sub-Saharan Africa. The same genetic shield that protected their bodies from one death was carried by their descendants into the conditions of another. And yet, the data said the exact opposite of what you'd expect next. Dr. Katarzyna Brick of 23andMe and Dr. David Reich at Harvard had already mapped this at the population level. In their 2015 landmark study in the American Journal of Human Genetics, Analyzing the genomes of more than 5,000 self-described African Americans, they found that on average, 20 to 24% of African American ancestry traces to European populations, with one very specific and very damning pattern. The European contribution arrived almost entirely through paternal lines. The European ancestry is in the Y chromosomes, not the maternal lineages. The fathers are European, the mothers are African. The genomic record of African American ancestry is not just a migration story. It is a crime scene. But stay with this, because inside that same genomic record, inside the sickle cell variant, inside the Benin and Bantu haplotypes that carry it, inside the specific frequencies that tell you not just that someone's ancestors came from West Africa, but which part of West Africa, there is a marker so geographically precise that it can name not just a region, but a specific population corridor that the Transatlantic Slave Trade Database confirms was actively trafficked in the 1780s. One mutation, one decade, one route, still present in living Americans who have no idea it's there. She is not in any census, not in any sale record, not in any church ledger. There is no date of birth, no date of death beyond the carbon dating of her bones, no record of her children's names, no record of who owned her or what she was called. But her genome is complete. Her mitochondrial haplogroup, the maternal lineage marker passed from mother to child in unbroken descent, places her, origin in Senegambia, consistent with the Wolof or Mandinka populations. Her relationship to 3 children buried nearby is confirmed by identity by descent segment analysis, the same method used to connect living relatives to consumer DNA databases, applied here to ancient bone. She was a mother 3 times, in a cemetery, in Maryland. In the 1790s, Dr. Iadao Harney and the Reich Lab documented this in the technical report archived before the science publication. The genomic relatedness analysis returned 3 confirmed first-degree relationships for this woman, 3 children, present in the soil beside her. And then there is what the children's genomes show. Among the 27 individuals at Catoctin, the technical report identified 3 individuals with Y chromosome haplogroups from European populations, specifically the R1b and R1a lineages, common in Ireland and Great Britain. Those are not the Y chromosomes of men brought from Africa. Those are the Y chromosomes of European men. And those chromosomes are in the children, not in the adult women. They are the paternal signal. The researchers stated this directly in the technical report. The pattern mirrors what is well documented in the historical and genetic literature as sex-biased admixture from rape and coerced sexual relations between white enslavers and enslaved black women. This isn't just ancient history. This is in your DNA right now. The genome does not record intent. It does not distinguish between consent and coercion. But the pattern, European Y chromosomes in the children, African mitochondrial DNA in the mothers, no European maternal lineages among the adult women. That pattern has one explanation, and the researchers named it. It is not a theory. It is the direct reading of what the chromosomes preserved. She had 3 children. She was buried near them. In a legal system that would not acknowledge she was their mother, in a country that erased her from every document, in a cemetery with no headstone carrying her name, the bones clustered together, the families held. And the European paternal signal in her children's chromosomes is a record of what was done to her, a record she carried in her body and passed forward through her children, who passed it forward again and again for 2 centuries, until a database in 2023 finally contained the machine sophisticated enough to read it as evidence. The families who enslaved the workers at Catoctin are documented. Their names are in the historical record. Some of their descendants still live in Frederick County, Maryland. And in the 23andMe database, among the 41, 799 living people matched to the Catoctin individuals. There are genetic relatives on both sides of that history. The genomic fault line that runs through that cemetery runs through living people today. It has never been resolved. The fault line runs through the living, not metaphorically, in DNA. And the question that nobody had ever quantified before is how many people that fault line actually represents. In July 2023, the same month as the Catoctin paper, Dr. Jaslyn Mooney of the University of Southern California and Professor Noah Rosenberg of Stanford University published a study in the journal Genetics that asked a question no one had thought to model before. Not what percentage of a Black American's genome is African or European, but how many distinct individuals, real people with their own parents and children and languages and names, does that percentage actually represent? Their computational model ran 14 generations of ancestry from 1619 to 1965, and it returned a number. A randomly selected African American born between 1960 and 1965 descends, on average, from 314 African ancestors and 51 European ancestors. 314 people, 51 people. not percentages, not fractions. People. There are three things this study revealed. The third one is the one that changes everything. The first, the peak number of African genealogical ancestors, the generation where the most distinct African individuals appear in a modern Black American lineage, occurs in birth cohorts from the early 1700s. That is the height of the trade. The bodies arriving in the greatest numbers in that decade became the most densely represented generation in the modern African American genome. The trade built a population, and the shape of that population is still readable today. The second, more than half of randomly selected African Americans have at least one European ancestor born after 1835. After 1835, close enough to the Civil War that this is not distant colonial history. Several of those 51 European ancestors, statistically, were alive during Abraham Lincoln's lifetime. And the third thing the study revealed, the one that changes everything about how you read a DNA result, comes in the next segment. But first, actually, I want to stop here for a second. In our last video, we looked at how conquest and forced population movement left permanent haplogroup signatures in European populations, marks still readable in the genomes of living people today. Drop in the comments, did that video change how you think about what a percentage on a DNA ancestry result actually means? Because what I'm about to tell you about those 51 European ancestors is directly connected to exactly that question. The third revelation is this. The African reference populations used to identify where in Africa those 314 ancestors came from were themselves reshaped by the same event being measured. Something happened to them. And when you understand what that something is, the entire architecture of African ancestry testing looks different. When a consumer DNA test tells you that your ancestry is 32% Yoruba or 18% Senegambian, it is comparing your genome against a database of living Yoruba and Senegambian people. And those databases are real. They are the best available science. But Dr. Raquel Flesques noted something in the Charleston Ancient DNA paper in PNAS in 2023 that most people in this space don't talk about. Some of the Charleston individuals, people buried in the late 1700s, before multiple generations of mixing had occurred, didn't match any contemporary African reference population with statistical confidence. Not because of poor preservation, because, as the paper stated directly, there was likely high population turnover in Africa as a result of the transatlantic slave trade. The populations that exist in West and Central Africa today are not identical to the populations that existed before the trade began. The trade removed people, but it also destabilized the communities left behind, reshaping marriage patterns, political structures, and demographic compositions across entire regions for generations afterward. What they found next changed everything they thought they knew about this, because despite this limitation, the broad strokes answer holds. Dr. Simon Gravel of McGill University, whose team analyzed 3,726 African Americans in their landmark PLOS genetics study in 2016, confirmed 1% African ancestry on average. The catoctin individuals connect to Senegambia and Congo-Angola. The Charleston individuals show diverse West African affiliations. The broad geography is recoverable. The ethnic specificity is where the signal gets uncertain, so in some sense, the system works. Until the final 20 seconds of that conclusion, when it doesn't. Here is what the limitation actually means for every African American who has ever received an ancestry breakdown by ethnic group. Those percentages, 32% Yoruba, 18% Senegambian, 4% Bantu, are estimates built against a reference population that was itself disrupted by the event being measured. You are not looking at a mirror. You are looking at a mirror that was broken and repaired and broken again. The broad shape is there. The specific face is not. Professor Fatima Jackson of Howard University has been analyzing mitochondrial DNA from the New York African Burial Ground for decades, the oldest and largest burial site of free and enslaved Africans in North America. Rediscovered in 1991 when construction workers in Lower Manhattan found 419 skeletal remains under an office building. Her analysis of the maternal lineages has found origins not just in West and Central Africa, but in East Africa, Southern Africa, and North Africa. A geographic range the standard narrative of the slave trade does not predict. The mirror shows more than one face. And this leads to something the genomic data from the Great Migration revealed, something about the structure of African American identity itself that turns out to be almost precisely backwards from what a century of historical scholarship assumed. Between 1910 and 1970, 6 million African Americans left the rural South for the urban North and West. The Great Migration, one of the most studied demographic events in American history, and the standard historical explanation, economic opportunity, Escape from Jim Crow violence, the pull of northern cities, is correct. The genomic data confirms it. But it adds a dimension that historians couldn't see, because it's only visible in DNA. Dr. Simon Gravel's team at McGill found something specific in the migration data. African Americans who left the South during the Great Migration had higher proportions of European ancestry, on average, than those who stayed behind. African Americans who had migrated north, approximately 16.5% European ancestry. Those who remained in the South, around 15.3%. African Americans already living in the North or West for longer, up to 25%. The direction is consistent. Higher European admixture, earlier migration, greater geographic reach. This discovery was made nearly a decade ago. Almost nobody talks about it until now. What this means is not comfortable. It means that the social hierarchies of skin color within Black America, the well-documented historical privileges extended to lighter-skinned African Americans in housing, employment, and access to northward mobility, are visible in the genome. The people with higher proportions of European ancestry as a group were able to move first, move farthest, and accumulate the advantages of earlier migration. The genetic record of the Great Migration is a map of colorism in DNA. Gravel's team also found what the migration left behind. African Americans who stayed in the South, who could not leave, or were not extended the marginal privileges that made leaving possible, retained the highest proportions of African ancestry on average. The most African genomes in America are concentrated in the communities that the Great Migration passed over. The communities most systematically denied access to 20th century economic opportunity are also genomically communities that preserved the most of what was brought across the Atlantic. The crimes encoded in the catoctin Y chromosomes and the patterns of the Great Migration are not two separate stories. They are one story, told across 150 years. And now the data does something that should not be possible. The most recent research, using imputation techniques developed specifically for low-coverage ancient DNA, has found genetic signals in the African American population that trace not just to the era of the slave trade, but to population movements within Africa that predate the trade by thousands of years. A migration that happened in West Africa 2 millennia before the first slave ship arrived in America. Still present, still legible, still here. That pre-slave trade signal is the Bantu expansion, one of the largest population movements in human history, beginning in West Africa, approximately 4,000 years ago, spreading Bantu-speaking peoples across sub-Saharan Africa over 2 millennia. The ancestors of the Congo people, the same group whose descendants were buried at Catoctin Furnace, were themselves the product of that expansion. The DNA does not begin with the Middle Passage. It begins in a West African homeland 4,000 years before the first ship. Dr. Katarzyna Breck and Dr. David Reich and the 23andMe research team confirmed this in their 2015 American Journal of Human Genetics study. The population structure within the West African ancestry segments of modern Black American genomes reflects not just geography, but linguistic affiliation, an echo of the Bantu expansion's language-linked spread, still readable in the chromosomes of people living in Baltimore and Detroit and Atlanta today. The genome goes back further than the trade. It always has. If you are African American, or if your ancestry traces anywhere through the African diaspora, through the Caribbean, through South America, through any community touched by the transatlantic trade, what I've just described is in your genome, not metaphorically. The Bantu expansion signal, the Senegambian or Congo-Angola maternal haplogroup, the sickle cell variant if you carry one copy, and in roughly 1 in 3 African American men, a Y chromosome that does not trace to Africa at all. All of it is there. The whole history, compressed into 4 billion base pairs, waiting for a machine sophisticated enough to read it. But here's what the machine cannot do. It cannot give them names. The researchers at Catoctin recovered everything: origin, family structure, health markers, disease susceptibility, relatedness to 41,799 living people. What they could not recover was the one thing that every legal system, every slave sale document, every plantation record, and every census before 1870 most thoroughly destroyed: who they were. The woman with the 3 children, she has a haplogroup, she has a confirmed family. She has living descendants carrying her mitochondrial DNA in their cells right now. She does not have a name. And the science that found everything else has no method for finding that. Before I say the last thing, I want to ask you something. If you've taken a DNA ancestry test, drop in the comments what it returned, not to compare results, but because after everything in this video, I want to know, does the number look different to you now? Does 32% Nigerian mean the same thing it did an hour ago? It shouldn't. If you want to go further, the Gullah Geechee DNA study, the New York African Burial Ground, the ongoing ancient DNA work at sites across the former Confederacy. It is all coming on this channel. Subscribe so you don't miss any of it. Catoctin furnace stopped producing iron in 1903. The last record mentioning the workers by any description was filed around 1850. For 173 years, the people in that cemetery had no scientific voice, until a machine finally learned how to read what the bones had been keeping. The bones remembered. We are only just beginning to listen.